So now let's finally discuss where all of that NADH, as well as FADH, which I need to discuss in a little bit more detail, is headed. It's headed to what's known as the electron transport chain. And the electron transport chain exists within the inner membrane of the mitochondria. So I've discussed the anatomy of the mitochondria already, but let's just emphasize a couple things here. This is the outer membrane, this whole thing here. Deep to the outer membrane is what's known as the inner membrane. Well, I'll do a different color. Is the inner membrane space, which is here. And then deep to the inner membrane space is the inner membrane itself. This would be the inner membrane here. Okay, so in between the outer membrane and inner membrane is the inner membrane space. Okay. The Krebs cycle was happening deep to the inner membrane, and that is within what's known as the matrix. All of this is the matrix. Deep to the inner membrane is the matrix. And that is where really all of the processes within the mitochondria occur. They occur in the matrix. Remember, the point was to get pyruvate as well as NADH that we created in the, in glycolysis. We wanted to get all of that into the matrix through the outer membrane and the inner membrane. And then we ran pyruvate ultimately through the Krebs cycle and produced a whole bunch of NADH as well as a little bit of FADH. And now we've got all of this NADH and FADH that we want to use in what's known as the electron transport chain. So where is the electron transport chain located? What you're about to see is the electron transport chain, which is a variety of proteins that are located, embedded within the inner membrane. Okay, this electron transport chain, there's not just one of them, there's many electron transport chains throughout the inner membrane. And deep to the inner membrane is the matrix where all of the NADH and FADH is. Okay, so we have these we can call electron transport chains embedded in the inner membrane, and now we have all this NADH and FADH in the matrix that can make contact with the electron transport chain, and now we want to discuss how that's used. All right, so to start off with, we've got lots of NADH as well as some FADH inside of the matrix. Right? The citric acid cycle or Krebs cycle takes place in the matrix, so we produce a lot of NADH and FADH in the matrix just naturally. Additionally, we looked at those transport mechanisms that the NADH was brought into the matrix additionally from glycolysis, which happened in the cytosol as well. Okay, so all of that NADH as well as a little bit of FADH that we created through the breakdown of this glucose molecule is within the matrix. Right, so we've got all this NADH and FADH, not as much FADH that's in the matrix, and it's all going to go to this, the electron transport chain. Let's start off discussing NADH. All of the NADH is going to go to what's known as cytochrome 1 or complex 1. And what's going to happen to NADH as it approaches complex 1 or cytochrome 1 is NADH itself is going to get oxidized. What's going to happen is it's going to lose its hydrogens and electrons, and it's going to go back to being NAD. And that NAD then can go back and participate in the citric acid cycle or in breaking or converting pyruvate into acetyl-CoA. All right, so that's the first step of the electron transport chain, is to have NADH become oxidized. And in essence, then, complex 1 becomes reduced because it has gained, ultimately, electrons. Okay. The electrons are passed to complex 1, and actually the hydrogens, minus their electrons, stay within the matrix. We call those protons or hydrogen ions, a hydrogen that no longer has its electron. Okay. So those electrons ultimately get passed to complex 1. And then what occurs is what's known as coenzyme Q comes over to complex 1. And what coenzyme Q does to complex 1 is ultimately it oxidizes complex 1 by becoming reduced itself. What I'm saying is it takes the electrons from complex 1. And by taking those electrons from complex 1, it takes the electrons then to complex 3. I know we skipped over complex 2. I'm going to talk about that in a little bit. Okay, so coenzyme Q takes these electrons to complex 3. And what complex 3 does to coenzyme Q is complex 3 oxidizes coenzyme Q, meaning that complex 3 takes the electrons from coenzyme Q. So complex 3 has now been reduced. Next, what happens? What we have here is what's called cytochrome C. Cytochrome C is going to come over to complex 3, and it is going to oxidize complex 3, meaning that it is going to take the electrons from cytochrome C, from complex 3, excuse me, and cytochrome C itself will become reduced. And then cytochrome C is going to go over to complex 4, and you can probably imagine what's going to happen here. Complex 4 is going to oxidize cytochrome C. It's going to take the electrons from cytochrome C, and complex 4 itself will become reduced, right, because it's gained electrons. So what you're seeing here is truly an electron transport chain. The electrons get passed from complex to complex to complex. And this is just a series of oxidation reduction reactions, so known as redox reactions, okay, which is what we've been talking about a lot throughout this chapter unit. So what is the point of all these oxidation reduction reactions that ultimately carry these electrons throughout the complexes? That's an important question that we need to answer. It would be inappropriate for me not to mention that complex 4 here, we're not going to discuss exactly how this happens just yet, but we're going to discuss it in detail a little bit later. Complex 4, which basically oxidized cytochrome C so that it could become reduced, meaning it gained electrons. Complex 4 will also get oxidized. A molecule is going to come along and it's going to oxidize complex 4, and that molecule then become reduced. Okay, that's important to understand, is that complex 4 will become oxidized as well. We're just not talking just yet about how that happens, but just make note of that. What you have to remember is as NADH approached complex 1, and NADH was oxidized by complex 1, meaning that it lost its hydrogens and electrons, the hydrogen ions, meaning the hydrogens without their electrons, stayed in the matrix, while the electrons then got passed down the complexes. Okay, so what's the point of passing those electrons down these complexes? Is that the passage of electrons down the complexes gives these complexes a form of energy to pump the hydrogen ions from the matrix into the inner membrane space. That's the point. Complex 1, complex 3, and complex 4 are all capable of pumping electrons. And as these electrons are passed from complex to complex to complex, this movement of electrons gives these complexes energy that allows them to pump these hydrogen ions. Again, I'm saying this repetitively, but it's important, from the matrix into the inner membrane space. So now you need to think about this in terms of time. What's going to happen over time is you are going to have an accumulation of hydrogens, particularly hydrogen ions, within the inner membrane space, right? Because as the electrons are passed between the complexes, that's going to keep allowing the complexes to pump hydrogen ions from the matrix into the inner membrane space. And as these hydrogen ions accumulate in the inner membrane space, that's going to do two things. It's going to create a chemical gradient, meaning that there's going to be a higher concentration of hydrogen ions in the inner membrane space than there is in the matrix. What do we know about chemical gradients? Well, molecules always want to move from a high concentration towards a low concentration. Besides just creating a chemical gradient, if we sequester these hydrogen ions more so in the inner membrane space than the matrix, we're also going to create an electrical gradient because we're going to move many more positively charged ions into the inner membrane space than the matrix. Right? So there's going to be a more positive charge in the inner membrane space and a more negative charge in the matrix. What do we also know? Positive ions like to move towards areas of negativity. Right? So that also is going to pull the hydrogen ions ultimately backwards. So by creating this gradient of hydrogen ions, we're creating what's known as an electrochemical gradient for the hydrogen ions to actually want to come back. They want to come back through a chemical driving force that just basically means from a high concentration to a low concentration, and also through an electrical driving force, right? The mitochondrial matrix is going to have a more negative charge, which is going to pull positively charged ions towards it. Okay, so that all has to have some purpose. Why do we create this gradient to make this collection of hydrogen ions want to come backwards? I have had some confusion in the past about what actually drives the pumping of these hydrogen ions. So let me try and clarify. When coenzyme Q, for example, comes over and it oxidizes complex 1, right? Complex 1 has these electrons that were given to it from NADH. Basically, complex 1 oxidized NADH and complex 1 became reduced. When coenzyme Q comes over and it oxidizes complex 1 and coenzyme Q becomes reduced, that's essentially, it's considered kind of the release of energy. That oxidation reaction where coenzyme Q removes the electrons from complex 1, that oxidation reaction results in the release of free energy that can be used to pump the hydrogen ions through complex 1 into the inner membrane space from the matrix. And that's true for all of these oxidation reactions. When complex 3 is oxidized by cytochrome C, that oxidation reaction results in the release of free energy, like the electrons being removed from complex 3 to cytochrome C, and that results in hydrogen ions being pumped. And then additionally, we haven't talked about this yet, but complex 4 also gets oxidized. And when it gets oxidized, again, that's the release of those electrons results in the release of free energy, and the complex can use that energy to pump the hydrogen ions from the matrix into the inner membrane space. Now let's get back to trying to answer how this electrochemical hydrogen ion gradient is going to drive the production of ATP. And the answer exists in this molecule here, which is known as ATP synthase. Okay? ATP synthase serves as a tunnel for these hydrogen ions to be able to come from the intermembrane space back into the matrix. And we know that these hydrogen ions want to come back through both the chemical and electrical driving forces. Okay? So if we create a tunnel for them to do so, they will. And what happens is, just as you might imagine, right, these hydrogen ions come back through ATP synthase. And as the hydrogen ions travel back through ATP synthase, again, we have ions traveling through this tunnel, which creates a type of energy that allows ATP synthase to rotate. And as ATP synthase rotates, it recombines inorganic phosphate with adenosine diphosphate and resynthesizes ATP. This is where the synthesis of ATP inside of the mitochondria actually occurs. It happens because of this hydrogen ion gradient that we create in the electron transport chain. Right? The hydrogen ions come back through ATP synthase, cause it to rotate, and as it rotates, it resynthesizes ATP by combining inorganic phosphate with ADP. Okay, so this is where ATP is made in the mitochondria, and that is the reason for bringing all of those hydrogens and consequently electrons to the electron transport chain. We stole those hydrogens and electrons off of the glucose molecule to, again, I can draw this up here, right? The main thing that we ripped off of that glucose molecule throughout all of these steps is lots of, ultimately, hydrogens and consequently electrons, right, in the form of NADH and some degree FADH. And we brought all of those hydrogens and electrons ultimately to the electron transport chain, which allows us to then power the electron transport chain, create this hydrogen ion gradient, rotate ATP synthase, and resynthesize ATP. So let's also now talk about oxygen, right? Because we all consume oxygen, and we say that we consume oxygen for the purpose of ultimately creating ATP. So clearly we're missing oxygen in this process, right? It's got to serve a function somewhere. So when you think of the electron transport chain as a whole, right, its purpose is to shuttle electrons throughout these complexes until they get to the fourth complex. And the shuttling of these electrons is what, of course, provides the energy to pump the hydrogens. But the idea is we need something to come and oxidize the fourth complex, right? Because this is where the electrons end up. Right? The electron transport chain is a series of oxidation-reduction reactions, and the fourth complex oxidizes cytochrome C and becomes reduced, and now we need to oxidize the fourth complex. And what will do that is oxygen. So what we know about atmospheric oxygen is it is two oxygen atoms, right? Combined together, they share electrons. That's why it's referred to as O2. We have oxygen that exists inside of all of our cells, right? Because ultimately we're going to use it in the electron transport chain for the production of ATP and what's known collectively as oxidative phosphorylation. That's what we've been discussing. So the question is, how and why is oxygen involved in this process? And the answer is oxygen has a very high affinity for electrons. It has a high redox or reduction potential, meaning it really likes to take electrons from other things and other molecules and become reduced. So all of these electrons, right, are traveling throughout the electron transport chain, and the electrons end up at complex 4. And what goes to complex 4? If you remember, I said complex 4 ultimately is going to get oxidized, right? And another molecule is going to get reduced. What goes to complex 4 and oxidizes complex 4 is oxygen, specifically one oxygen atom. Atmospheric oxygen is made up of 2 oxygen atoms bound together, and what we're talking about here is molecular oxygen, which is an oxygen atom by itself. That's why over here it says 1/2 of O2. It's because it's one oxygen atom headed towards complex 4. And this oxygen atom has a very high reduction potential. So what it's going to do is this oxygen atom is ultimately going to steal 2 electrons from complex 4. It's going to remove those electrons from complex 4, which is very important, as we'll discuss, because otherwise these electrons would build up at complex 4 if oxygen didn't take these electrons, and this whole system would be backed up. But that's, again, for a conversation for a little bit later from now. So this oxygen takes 2 electrons, and then it combines with 2 hydrogen ions, and that forms a molecule of H2O. And that is a byproduct of oxidative phosphorylation. And that is how the electrons are removed from complex 4. It's from oxygen, which has a high affinity for electrons, stealing the electrons, oxidizing complex 4 and becoming reduced, and then combining with 2 hydrogen ions to form water, H2O. And when complex 4 is oxidized, if you remember, that results in the release of free energy when complex 4 is oxidized, and that's actually how the last bit of hydrogen ions gets pumped from the matrix into the inner membrane space. So that's the last step in the electron transport chain. It's oxygen coming to complex 4 and oxidizing it. And just to make a point, as you travel throughout the electron transport chain, each protein has a higher redox or reduction potential than the next. For example, complex 1 has a higher reduction potential than NADH. That's how complex 1 oxidizes NADH and becomes reduced. Coenzyme Q has a higher reduction potential than complex 1. That's how coenzyme Q oxidizes complex 1 and becomes reduced. Complex 3 has a higher reduction potential than coenzyme Q. That's how complex 3 oxidizes coenzyme Q and becomes reduced. Cytochrome C. I don't know if I need to finish all of this for you, but cytochrome C has a higher reduction potential than complex 3. So cytochrome C oxidizes complex 3 and becomes reduced. The same thing is true for complex 4 versus cytochrome C. And then lastly, oxygen is last in line, and oxygen has the highest redox or reduction potential. And that's how the electrons are finally removed from complex 4. It's essential that every protein in line has a higher reduction potential than the last one. Otherwise, the electrons wouldn't keep moving down the chain. And hopefully now you can see why oxygen is so important. If you don't have oxygen, your cells really can't produce any, ultimately, ATP aerobically, because you're going to see fat is made in a different process, but uses the electron transport chain just like glucose does. So without oxygen, you don't make ATP aerobically, and your cells die because you need that oxygen, which has the highest reduction potential, to remove those electrons from complex 4. If you don't remove those electrons from complex 4, the whole system backs up, you don't produce any more ATP, and the cell dies. So that's why oxygen is so fundamental to human life. It's because we need it for the production of ATP within our cells, because of its reduction potential. All right. We're going to talk about that in more detail and be more specific about how not enough oxygen would drive a reduction in the hydrogen ion gradient and why that wouldn't result in any ATP production. But maybe you can think through some of that in advance and realize why oxygen is so fundamental to this process. Now let's quickly discuss how much ATP each NADH molecule would make, and also FADH molecule. So before I talk about the amount of ATP that's created from NADH versus FADH, I want to provide some important vocabulary that should hopefully make sense now based upon the things that we talked about. Okay, there's this term which is in mitochondrial membrane potential. Okay, what is the mitochondrial membrane potential? A membrane potential is a charge. Okay, we're going to talk about membrane potentials a lot more in the future, but a membrane potential is a charge. And a membrane potential is a charge that typically exists across a membrane. That's where membrane potential comes from. Okay, membrane potential, charge across a membrane. Okay, so when we say mitochondrial membrane potential, what we mean is the charge that exists across the inner membrane. Okay, so how is there a charge that exists across the inner membrane? Simple. The inner membrane space is positive due to all of the hydrogen ions that exist in the inner membrane space, and the matrix is negative due to there being less hydrogen ions in the matrix. All right, so the inner membrane space is positive. We talked about this. The matrix is negative. So there is a charge that exists across the inner membrane. A charge that exists across a membrane is by definition a membrane potential. So the mitochondrial membrane potential is the charge that exists across the inner membrane. The inner membrane space being positive and the matrix being negative. So the next part of this is recognizing that the mitochondrial membrane potential is, or certainly creates, what's known as the proton motive force. You can say it either way. I mean, the mitochondrial membrane potential is ultimately the proton motive force. So what is the proton motive force? Let's just simplify this. Okay, the proton motive force is just the desire or the drive for the hydrogen ions to come from the inner membrane space back into the matrix through ATP synthase. That's what the proton motive force is. Okay, the pull for hydrogen ions to come from the inner membrane space back into the matrix. So now if you think about what creates the proton motive force, what creates the proton motive force, this desire for hydrogen ions to come from the inner membrane space back into the matrix? Well, what creates the proton motive force, or is the proton motive force, is the mitochondrial membrane potential. The fact that the inner membrane space is positive and the matrix is negative, that is what pulls the hydrogen ions from the inner membrane space back into the matrix. Now, partly they want to come back just through a diffusion gradient, but the biggest of the two, the biggest factor, is the mitochondrial membrane potential. That's the main thing that pulls these hydrogen ions from the inner membrane space back into the matrix. So, to summarize, the mitochondrial membrane potential is the difference in charge that exists across the inner membrane. The inner membrane space is positive, the matrix is negative. The proton motive force is the desire or the pull of the hydrogen ions from the inner membrane space back into the matrix. What creates the proton motive force, or you'll oftentimes see it referred to as being the proton motive force, is the mitochondrial membrane potential. The fact that the inner membrane space is positive and the matrix is negative, that is the mitochondrial membrane potential. And that membrane potential creates or is the proton motive force, the thing that pulls the hydrogen ions from the inner membrane space into the matrix. So let's get back to comparing ATP production by NADH and FADH. All right, so how much ATP does each NADH produce compared to FADH? So NADH, as you know, comes to complex 1 and is then oxidized by complex 1. So the electrons which are brought to the electron transport chain by NADH, travel from complex 1 to complex 3 to complex 4. So the electrons from NADH are ultimately going to drive the pumping of electrons, the pumping of hydrogen ions by complex 1, complex 3, and complex 4. You think, well, why would that be the case? Well, because the electrons from NADH are ultimately given to 1, passed to 3 by coenzyme Q, and then passed to 4 by cytochrome C. So those electrons from NADH are going to contribute to the hydrogen ion pumping of complex 1, 3, and 4. Okay. So it contributes quite a bit, each NADH molecule, to the hydrogen ion gradient. What about FADH? I did not mention this to you quite yet because I didn't think it would be helpful if you didn't know the electron transport chain to say this yet. But complex 2 of the electron transport chain, which we have not discussed yet, is actually succinate dehydrogenase from the Krebs cycle. The Krebs cycle is not actually some perfect loop that exists inside of the matrix, despite it being drawn that way. The enzymes are all over the place inside of the matrix, driving one reaction after the next. And complex 2 is actually that succinate dehydrogenase enzyme from the Krebs cycle. If you don't remember, go back and take a look. And what succinate dehydrogenase does is it results in the oxidation of one of the intermediates to form FADH. Some hydrogens and electrons are stolen off of the intermediate, driven by complex 2, which is the enzyme succinate dehydrogenase. And FADH takes those hydrogens and electrons and becomes FADH. And FADH actually exists within complex 2. So it's kind of a minor fact, but I want to be as accurate as I can be. And then what happens is complex 2 has its electrons taken from it, or we could say complex 2 is oxidized by coenzyme Q. So if we really want to be incredibly specific, complex 2 is succinate dehydrogenase, and it's involved in oxidizing succinate to fumarate. Okay, so complex 2 oxidizes succinate to fumarate, and that results in the production of FADH. All of this we collectively say happens within complex 2. FADH within complex 2 is then oxidized by coenzyme Q. Right? Coenzyme Q becomes reduced, and then it carries the electrons to complex 3, which oxidizes coenzyme Q, and then complex 3 becomes reduced. What's important to note here is that complex 2 doesn't pump any hydrogen ions at all. Okay? The electrons from complex 2 are really coming from FADH within complex 2, and coenzyme Q picks up those electrons, and in oxidizing FADH within complex 2, that doesn't result in any hydrogen ions being pumped through. Complex 2 is not a transmembrane protein. It doesn't drive the translocation, we could say, of hydrogen ions from the matrix into the intermembrane space. All that happens is the electrons from complex 2, which coenzyme Q is really taking from FADH, are brought to complex 3, and complex 3 then oxidizes coenzyme Q to become reduced. Then finally, cytochrome C will oxidize complex 3 and become reduced, and that will result in the pumping of some hydrogen ions. At the end of the day, FADH within complex 2 only results, its electrons only contribute to hydrogen ion pumping within complex 3 and complex 4. versus NADH. The electrons from NADH result in hydrogen ions being pumped within complex 1, complex 2, and complex 3. So the point of saying all of this is that NADH, since the electrons from NADH contribute to the pumping of hydrogen ions within complex 1, 3, and 4, and the electrons from FADH only result in hydrogen ions being pumped within complex 3 and complex 4, each NADH molecule contributes more to the hydrogen ion gradient than each FADH molecule does. Hydrogen ions coming back through ATP synthase causes ATP synthase to rotate and then resynthesize ATP. Okay, if NADH contributes more to this gradient than FADH does, then of course NADH contributes to a greater production of ATP than each FADH molecule does. So without getting into all the specifics of how many hydrogen ions are pumped by each complex, what I can just tell you is this. What we know is that for each NADH molecule, it contributes enough to the hydrogen ion gradient that about 2.5 ATP are made for each NADH molecule. Compared to FADH, which contributes less to the gradient, and by consequence only results for each FADH, only 1.5 ATP are formed. And again, that's because it contributes less to this hydrogen ion gradient, contributes less to ATP synthase rotating, so less ATP are created for each FADH. So the process we have gone through now is how ATP is made aerobically with oxygen. Right? You know oxygen's job is to take electrons from complex 4. It oxidizes complex 4 so that it can be reduced and then combined with hydrogen ions to form water. But this is why this is called aerobic ATP production. And specifically, this aerobic ATP production is collectively known as oxidative phosphorylation. You think about what does oxidative phosphorylation mean? You can break this down pretty easily. Oxidative. There's a whole variety of oxidation reactions, right? NADH is oxidized by complex 1. Complex 1 is oxidized by coenzyme Q. Coenzyme Q is oxidized by complex 3. Cytochrome C oxidizes complex 3, complex 4 oxidizes cytochrome C, and oxygen oxidizes complex 4. So oxidative, right? A series of oxidation reactions that results in the phosphorylation of ADP back to ATP. And to think about that more entirely, these oxidation reactions allow for hydrogen ions to be pumped into the inner membrane space, creating a hydrogen ion gradient, right? And then the hydrogen ions can come back through ATP synthase due to the both chemical and electrical pull for the hydrogen ions to come back, and the ATP synthase can then rotate. So the oxidative portion creates this hydrogen ion gradient, and then the hydrogen ions coming back through drives the phosphorylation of ADP back to ATP. So that's why it's called oxidative phosphorylation. And oxidative phosphorylation is an aerobic process, meaning that it's dependent upon oxygen, and hopefully you can see why. So we can compare oxidative phosphorylation, this process, to substrate level phosphorylation, like what happened in glycolysis. With substrate level phosphorylation, you've got a substrate, one of the intermediates in glycolysis. And from that substrate, a high energy phosphate is stolen, and that high energy phosphate being taken off of the intermediate directly results in the resynthesis of ATP, or the phosphorylation of ADP back to ATP. So that's substrate level phosphorylation, where you resynthesize or phosphorylate ADP back to ATP by taking the phosphate directly from a substrate. And then you have this process, oxidative phosphorylation, which is clearly different for the reasons that we just discussed. Think about oxidative phosphorylation, put them together, right? It's a completely different process to make ATP than substrate level phosphorylation. You're relying on oxidation reactions to create a hydrogen ion gradient to phosphorylate ADP back to ATP. And of course, it's important to mention in the Krebs cycle, you also do get one step of substrate level phosphorylation when GTP is made, and then that donates its phosphate to ATP. But there's a distinct difference too. The substrate level phosphorylation in glycolysis, you can run that independent of oxygen if you run what's called anaerobic or fast glycolysis. In order for substrate level phosphorylation to occur within the Krebs cycle at a continued rate, you actually need the electron transport chain, oxidative phosphorylation, to be running at a good rate. Otherwise, that substrate level phosphorylation at the level of the Krebs cycle is meaningless. And hopefully our coming discussion here will make sense of why that is. You could never rely on the substrate level phosphorylation of the one step within the Krebs cycle to meet a very high ATP demand, because it's actually dependent upon this system running sufficiently. While you could be somewhat dependent upon the substrate level phosphorylation that happens in anaerobic glycolysis, because anaerobic glycolysis is completely independent of all of this, as we will discuss. I'm about to discuss with you how much ATP is made from one glucose molecule if it's broken down aerobically through oxidative phosphorylation. Please know I will not ask you a question on any exam about numbers. I'm doing this just for clarity. I'm not going to ask you to explain how a given glucose molecule produces roughly 32 ATP. It's good for you to know because it'll test, how well you're understanding these concepts. But that's not the point of the exam. I do want you to know these processes by which glycolysis produces ATP via substrate level phosphorylation, how and why anaerobic glycolysis happens, and then also how ATP is made through aerobic or slow glycolysis. There will never be a question on the exam about one intermediate being converted into the next, like what happens to fumarate or what enzyme is involved in converting fumarate. You will never see anything like that. You would see a question like, what's the function of the Krebs cycle? That is big, right? Because what the Krebs cycle really does is it oxidizes intermediates to create lots of NADH and FADH. What's the function of NADH and FADH? Well, it goes, FADH is technically really already in the electron transport chain because of complex 2, but FADH and NADH are used in the electron transport chain. How and why are they used? Well, the electrons are pumped throughout the chain, the complexes, to create a hydrogen ion gradient, so the hydrogen ions can come back through ATP synthase, rotate and create ATP. How is oxygen used, right? It has a high reduction potential and it removes the electrons from complex 4. It's the big picture conceptual things that I want you to know. I try to do my best to be specific in the goals or the objectives that I give you, that's the case. But I still get questions and I totally understand it. And if you still have questions, by all means, email me so that I can clarify it to you. Or if there's an objective you want clarification on, but there's not going to be step-by-step questions about glycolysis or the Krebs cycle. Not going to happen. Doesn't mean I don't want you to know the overall purpose of glycolysis, fast glycolysis or anaerobic glycolysis, slow glycolysis, or aerobic glycolysis as it's sometimes called. That would include knowing the purpose or function of the Krebs or citric acid cycle, as well as the electron transport chain. You want to know the big picture component of these things and how they operate, not the fact that malate is converted into oxaloacetate by malate dehydrogenase. Sorry. Those are not the kind of questions you'll see. All right. So let's continue on and discuss, and you can test your knowledge here about how much ATP is made through these two processes. So let's go ahead and talk about how much ATP can be made from a glucose molecule when we break it down aerobically with oxygen through this process of oxidative phosphorylation. To start off with, we run the glucose molecule through glycolysis, and through substrate level phosphorylation, we get 2 ATP. We actually get 4, but the net gain is 2, so we use 2 at the beginning. Additionally, through glycolysis, through these oxidation reactions, we end up with 2 NADH, which then through that shuttle mechanism enter into the mitochondria, go to complex 1 most of the time, and contribute each to the production of 2.5 ATP, giving us a total of 5. Also through glycolysis, we get 2 pyruvate molecules which enter into the mitochondria and are each then oxidized to acetyl-CoA. Each time pyruvate is oxidized to acetyl-CoA, we get 1 NADH. That happens 2 times. Each go to the electron transport chain and contribute then to 2.5 ATP, giving us a total of 5. So up to this point, we have 12 ATP. Now, what will happen next is each one of those acetyl-CoA molecules that was created will go around the Krebs cycle. So we get 2 rounds of the Krebs cycle. Each round of the Krebs cycle gives us 1 ATP. So we get 2 ATP from the Krebs cycle through substrate level phosphorylation. Each round of the Krebs cycle also gives us 3 NADH, which then translates into a total of 6 NADH due to 2 rounds. Each NADH will go to the electron transport chain and contribute to the production of 2.5 ATP. That happens 6 times, so another 15 ATP are created. Next, for each round of the Krebs cycle, we get 1 FADH, so that gives us a total of 2, and through its contribution at complex 2, 1.5 ATP is created for each FADH, giving us a total of 3 more ATP. And you add all of this up, and you see that 20 more ATP are created. So what this tells us is when we break a glucose molecule down with oxygen, aerobic or slow glycolysis as it's called, utilizing oxidative phosphorylation, we get about 32 ATP. Compare that when we rely on breaking the glucose molecule down with no oxygen, called anaerobic or fast glycolysis, and we only get 2 ATP from the glucose molecule. So you can see you get a lot more ATP from a glucose molecule if you break it down aerobically, utilizing oxidative phosphorylation, than if you break the glucose molecule down anaerobically without oxygen and you rely entirely on substrate level phosphorylation. As we're going to talk about in our next discussion, if you can bypass oxygen, you can make ATP a lot faster, most likely, than if you rely, I shouldn't say most likely, definitely, than if you rely on oxygen for ATP production. So yes, aerobic or slow glycolysis, which utilizes oxidative phosphorylation, makes a lot of ATP per glucose molecule, but as we're going to talk about, it's probably limited by oxygen to a degree, the rate at which it can produce ATP, so it produces it slower. Comparatively, anaerobic or fast glycolysis is not dependent upon oxygen, and while it only makes 2 ATP from a glucose molecule, it can make those ATP very fast. Okay? So the question is, in what circumstances, and you should already know this to a degree, but in what circumstances is the cell going to preference one system versus the other? And ultimately they use them both, but what we want to look at is, and by that I mean they use them both simultaneously, but what we want to look at is when is one or the other preferred in a given circumstance, and ultimately what will cause the transition between one or the other of these systems. That's what our next conversation is going to be about.
So one last thing I wanted to talk to you about, another sort of vocabulary related thing, is what's called uncoupled oxidative phosphorylation. Okay, you have all the tools, I think, that are needed to understand what uncoupled oxidative phosphorylation is. Okay, so let's first talk about what coupled oxidative phosphorylation is. It's actually exactly what we've been talking about this entire time. What regular oxidative phosphorylation results in is the coupling of two reactions. Okay, in chemistry, you have exergonic, endergonic coupling. One reaction is coupled or allows for the other reaction to occur. Okay, hydrogen ions moving through ATP synthase is a form of a reaction. The movement of hydrogen ions from the intermembrane space back into the matrix releases energy. That's actually a reaction itself. That's reaction one. Reaction one is the movement of the hydrogen ions from the intermembrane space back into the matrix through ATP synthase. And that reaction releases energy. And that reaction is going to be coupled to another reaction. And you can probably figure out what the second reaction is. The second reaction is ATP synthase taking ADP and phosphorylating ADP back to ATP. Okay. The first reaction allows for the second reaction. They're coupled to each other. Without the first reaction, you don't get the second reaction. They're coupled. That's what the term means. You 100% all of the time need the first reaction to occur in order for the second reaction to occur. They're coupled to each other, so without each other, they don't happen. I should say that differently. If the first reaction isn't coupled to the second reaction, you don't get the ultimate desired end product, which in this case is ATP. Okay. So just to recap, this is important for the second part. These two reactions are coupled to each other. The movement of the hydrogen ions through ATP synthase is the first reaction that releases heat, and it allows for the second reaction, which is the creation of ATP by ATP synthase. I'm clearly getting ahead of myself. I meant to say the movement of the hydrogen ions through ATP synthase is the first reaction, and it releases energy, not heat. That's coming later on. It releases energy that ATP synthase then uses in the second reaction to create ATP from ADP. Now, what uncoupled oxidative phosphorylation will do is it will uncouple the two reactions. I mean, it's right in the name, right? The movement of the hydrogen ions through ATP synthase is the first reaction that allows for the second reaction of creating ATP by ATP synthase, right? Those are coupled reactions. Uncoupled oxidative phosphorylation uncouples those two reactions. How does it do it? It does it through this protein here, which is uncoupling protein 1, UCP1. What UCP1 is a hydrogen ion pore. It allows the hydrogen ions, which are in the intermembrane space, to get back into the matrix. And those hydrogen ions no longer have to go through ATP synthase anymore. Right? They don't have to go back through ATP synthase. They can go through uncoupling protein 1, UCP1. And immediately you can observe what's happened here. Right? We've uncoupled the two reactions related to hydrogen ion movement from the intermembrane space back into the matrix and the creation of ATP by ATP synthase. Those two reactions are no longer coupled to each other, thanks to UCP1, uncoupling protein 1. It's given the hydrogen ions another means to get back into the matrix. So ATP will no longer be created by ATP synthase because you need the first reaction, hydrogen ions moving back into the matrix, in order for the second reaction, ATP being created by ATP synthase, to occur. Okay? So we don't get ATP production anymore in uncoupled oxidative phosphorylation. What do we get, though? Because hydrogen ions moving from the intermembrane space back into the matrix, that's the first reaction, and in that first reaction, energy is always released. But if it's not coupled to the second reaction of making ATP by ATP synthase, what happens to the energy of the hydrogen ions moving from the intermembrane space back into the matrix? And the answer is the energy is released as heat instead of the energy being used to create ATP by ATP synthase. So by uncoupling the reaction between hydrogen ions moving back into the matrix and ATP synthase creating ATP, you've allowed for the movement of hydrogen ions back into the matrix. That energy, instead of being instead of that energy allowing for the second reaction to occur, that energy is just simply released as heat. This happens in brown fat in babies and in bears, and it also contributes to what's known as non-shivering thermogenesis, which we think actually can happen in skeletal muscle too, becoming an interest in exercise physiology. The electron transport chain continues to operate the normal way that it does in the mitochondria. But the difference is the hydrogen ions moving back into the matrix is used for the production of heat rather than for the production of ATP by ATP synthase. And it allows for the human body to be heated, or in some specific animal during hibernation, for example. So my hope is that the term uncoupled oxidative phosphorylation makes some sense to you.